In this video, we will show what different levels of color blindness are like. It is number 4 in our series about color blindness. The rich level of color vision that most of us enjoy is based on having 3 fully functioning color cones, resulting in what is called trichromatic color vision. We also have rods for vision in low light, but they don't contribute to color vision. Things, of course, don't always go as planned. If only one cone is functioning, the world would look like this, but this is pretty rare. Much more common is some degree of red-green color deficit, occurring in approximately 1 in 8 men and 1 in 50 women. How the different deficits are likely to appear is the subject of this video. Details on the mechanism and mode of inheritance are covered in the previous videos. As we did in our first 2 videos, we begin our story with John Dalton, who is known both for the development of basic atomic theory and is one of the most famous examples of color blindness. Here is the color spectrum as someone with normal color vision sees it. Dalton, looking at the same spectrum of colors, described how he saw it. Quote, "I found that persons in general distinguish 6 kinds of color in the solar image, namely red, orange, yellow, green, blue, and purple. I see only 2, or at most 3 distinctions. These I should call yellow and blue, or yellow, blue, and purple. My yellow comprehends the red, orange, yellow, and green of others, and my blue and purple coincides with theirs." From that description, a non-colorblind person can get a pretty good idea of what the world looks like to someone with his particular kind of color blindness. As a scientist, Dalton was curious. What was different about his eye? He guessed it was because the fluids inside his eye were tinted blue. He was curious enough that he directed that his eye be examined after his death in 1844. The fluids and media were found to be clear, not blue. There's more to the story. Some of his eye tissues were preserved so that in 1995, when the technology was available, gene testing was done. It showed that the green or M cone pigment was absent. That put him in the category of red-green color blindness. In technical terms, because he used 2 color cones, that made him a dichromat, and more specifically, missing the green cone made him a deuteranope. We will explain the terms later on, but I want to note here that that makes his kind of color blindness a bit more severe than the typical kind. This was such a famous example that color blindness often went by the name Daltonism. As we said at the outset, the rich level of color vision that most of us enjoy is based on having 3 color cones. Rods are for vision in low light and don't contribute to color vision. Color deficiency usually occurs in one of 2 levels. The simplest to understand is when one entire cone class is missing. Actually, it is possible to miss more than one cone class, but that is rare. For example, if the green cones are missing, as we saw with Mr. Dalton, this results in a deficit in red-green color vision. In terms of the color spectrum, you are trying to discern all the colors with just 2 cone pigments. The second level of color deficiency is a bit more complicated. Some years ago, the theory was that all the cone classes were present, but one of the cone pigments was just not functioning properly, so perception of that particular color was weak. This is an example of a theoretically weakly functioning green cone. A good deal of research starting in the 1980s revealed a more complex explanation. This level can be better described as resulting from a hybrid or chimeric cone pigment. I'm exaggerating a bit with the yellow color to make the point. It actually looks more like this. One version comes with a red and a red-like cone pigment, and the other version comes with a green and a green-like cone pigment. Depending on how far apart the normal and hybrid pigments are separated, determines how much color sensitivity one will have. This can range from near normal to a full deficit, behaving just like a dichromat. With that as background, let us take a step back, broaden our view, and take a look at all the different levels of color deficiency, from monochromat to anomalous trichromat. Here again is our standard complement of cone pigments. In humans, there is a rare condition in which a person has only rods and no cones. The term for this is a rod monochromat, one color. This results in a grayscale vision like this in dim light. You may remember that there are only cones in the fovea, the very center of the retina that supplies fine detail vision. So, without cones, the detail part of the central vision is missing. Also, they lack the ability to hold steady fixation. Both of these factors directly result in poor acuity, which I've attempted to show here. Also, someone with only rod vision can't function well in bright light, because the rods are bleached out. Somewhat like this, maybe worse. They only function in dim light. There are some animals, however, in which this is their standard condition. Deep under water, below about 1,000 meters, all is essentially darkness. Fish that live at these depths have made several visual adaptations, including the use of only rods. Next, what if you had only one type of cone? You would be called a cone monochromat. What color do you think you would see? Trick question. Whichever cone you have, the term color doesn't have much meaning in this situation. You would only be able to tell differences in brightness. That means you would see the world in grayscale, like a black and white TV. In humans, being a cone monochromat occurs as a rare condition. There are two varieties. If either the L or M cones are functioning, then vision is grayscale with relatively good acuity. However, if only the blue cones are functioning, then that's less good. Why? This shows the concept of the mosaic of cones in the fovea. You will notice that there are no blue or S cones in the center of the fovea, only red and green ones. If you have either red or green cones, you have some foveal function and acuity can be relatively good. But if you have only blue cones, then, like the rod monochromat, there are no cones in the fovea and visual detail is poor, maybe a little better than the rod monochromat. There are animals for which being a cone monochromat is the standard. Among terrestrial mammals, owl monkeys, which are nocturnal, have only one cone, sensing in the middle to long wavelength range. Many marine mammals, including whales, dolphins, and seals, have only one cone. What if you had two types of cones? This person is a dichromat. As an example, say the red cone is missing, the blue and green ones are working fine. Then he or she is called a protenope. Missing the red cone means all the color information comes from the blue and green cones. In the simple cone color model, you might expect the resulting vision to be a palette of blue and green, somewhat like this. However, it doesn't end up that way. The final vision is more like this, because of the color opponent process. Color information is separated into red, green, and blue-yellow channels. In this case, the result is a blue-yellow palette. By far, the most common combination of cones among mammals is to have both S and L pigments. This includes dogs and cats, as well as pigs, cows, horses, and elephants. So their vision is like this, which we showed before. And this is how Dalton described his vision. Here is what the spectrum looks like to the three different types of dichromats. The protan and deutan, lacking red and green respectively, fall into the category of red-green deficiency, and the visual result is quite similar. The tritan, lacking blue, falls into the category of blue-yellow deficiency, and the result is quite different. What we have just described are the dichromats, people who have two functioning cones. The more common level of color deficiency are people who have three functioning cones. Yes, three cones should give trichromatic color vision, but it is not the standard set of three cones. But as we said in the beginning, one is a hybrid or chimeric cone. These people are trichromats, but anomalous trichromats. Color deficit ranges from mild to severe, the latter being like the dichromats. Simulation of color vision deficiency for dichromats is relatively straightforward, but for anomalous trichromats it's more difficult. Think about it. It is not just about missing a cone, it is about a shift in cone sensitivity added to the result from the color opponent system. Here is a notable paper on color simulation, which takes into account both cone color response and opponent color processing. The quantitative relation between cone input and opponent channel output is shown in this diagram, taken from the earlier work of other researchers, Ingling and So, referenced at the bottom. Based on this model, the authors attempted to make a more physiologically faithful reproduction of the likely color vision result. Remember, the spectrum of the hybrid cones was shifted by varying amounts. In their model, they input a shift of various amounts, then they simulated levels of resulting color vision for both protanomaly and deuteranomaly, shown here. On the left is the normal color spectrum. To the right are the results of increasing color deficit. At the far right is the extreme level, which is equal to a dichromat. In this illustration from their article, they are comparing their results with another model by Yang, which did not account for the opponent color process. If you look closely, you will see some subtle differences. In this illustration, they have applied their model to the artificial colors of a brain scan. For one more example of color simulation, I've included this from another website, which is cited at the bottom. I'm not sure what method they use, but I thought it gave a nice example of the concept of progression of color loss. This is an example of deuteranomaly and deuteranopia. Now, we have spent quite a bit of time talking about the different kinds of color deficiency. The next question is, how often do they happen? This chart shows the percent of males and females with one entire cone class missing. These are the dichromats. You can see that in males, missing a red or a green cone occurs about 1% of the time. Missing a blue cone is much less common. This chart shows the trichromats, those with two normal cones and a hybrid red or green cone or a defective blue cone. Again, in males, the red-green deficit is much more common. If you add those up, red-green deficits occur in about 8% of males, which is about 1 in 12. Though color deficiency has been known for a long time, it is only in recent years that designers are paying attention to color choices in graphics and web page layout. Here is an example of a website which reviews color blindness and proposes useful color choices. There are numerous articles and websites about color design. I just chose this as one example. Here is a review of the videos in our series about color blindness. In the first video, we presented a review of color vision and basic genetics, including location of the color genes on different chromosomes. Because the red and green genes are located next to each other on the X chromosome, their inheritance is X-linked, and they are subject to frequent recombination. In video 2, we looked at how recombination resulted in lost color genes, or created hybrid or chimeric genes, which are the basis of inherited red-green color deficiency. In the third video, we used color science to understand the resulting color deficiency in a more technical way. And in this last video, we tried to simulate what color deficiency looked like, and to use the previous information to understand why it appeared that way. If you want to learn more, here are excellent selected references.